Transcription is the process of copying the information from DNA and creating a messenger RNA out of it as a set of instructions to make proteins. Transcription occurs in the nucleus. DNA nucleotides are what make up DNA, and they are made from a phosphate group, deoxyribose sugar, and a nitrogen-containing base that is either adenine, thymine, guanine, or cytosine. RNA nucleotides make up RNA, and they are made of a phosphate group, a ribose sugar, and a nitrogen-containing base that is either adenine, uracil, guanine, or cytosine. The first thing that happens is transcription factors binding to the DNA so that RNA polymerase can bind. Transcription factors also help regulate transcription. Next, RNA polymerase, the enzyme that controls the transcription process, binds to the DNA at the TATA box, which is a promoter, meaning it is what tells the enzymes where to begin due to its sequence of bases. The RNA polymerase will move along the DNA strand starting at the promoter and unwinding the DNA as it goes so that it can copy just one strand. The RNA polymerase moves from the 5' end to the 3' end. After DNA is unwinded, RNA polymerase starts to read the DNA, and RNA nucleotides are brought in and temporarily bind to the DNA template strand, creating a DNA-RNA hybrid complex. The DNA template strand is the strand that is being copied, so an RNA strand can be created. RNA elongation is when more amino acids are brought in to bind to the DNA strand, allowing the RNA strand to keep growing. Eventually, RNA polymerase will reach the termination site, which is another sequence of bases. At this point, the enzyme stops copying the DNA, and the RNA strand is spliced at the end. Next, a 5' cap is added to the 5' end of the RNA, and a 3' tail is added to the 3' end, so that the RNA strand does not degrade. After capping, splicing occurs so that introns can be cut out. Introns are sequences that do not carry any coding, and they interrupt the genes. After they are cut out, the exons, the sequences that do carry coding, bind together. At the end of transcription, a messenger RNA has been made. A messenger RNA is a copy of the DNA strand, and it is used as instructions to make proteins in translation. Throughout the process of transcription, mutations can occur. Mutations are caused when bases are either changed, added, or taken out, and they can affect the DNA to be different than normal, meaning it might not function properly.
Translation is the process of using messenger RNA as a template to make proteins. Translation occurs in the cytoplasm. RNA nucleotides make up RNA, and they are made of a phosphate group, a ribose sugar, and a nitrogen-containing base that is either adenine, uracil, guanine, or cytosine. A codon is any sequence of bases made of 3 nucleotides and can have multiple different purposes. Messenger RNA is the copy of a DNA strand made in transcription and is used as a template to make proteins and translation. To start translation, a ribosome binds to the messenger RNA. Like the RNA polymerase and transcription, the ribosome controls translation. The ribosome starts translating sequences of 3 bases. The start codon is the first codon to be translated. When the ribosome reads the codons, a transfer RNA carries anticodons to the ribosome. Anticodons are sequences of 3 bases that are complementary to the codons of the RNA. The transfer RNA binds to the ribosome at the A site. The anticodon the transfer RNA is carrying binds to its complementary codon at the P site to create an amino acid. Transfer RNA leaves the ribosome at the E site. Amino acids are the anticodon, and they function as the building blocks for proteins when in a chain. Elongation of amino acids occur when anticodons are added to the amino acid. After the amino acid chain is finished being made, translation is done, and the protein breaks off from the messenger RNA. The amino acid chain is also called a protein, which makes up structures in the body and performs many functions in the cells. Like in transcription, the bases can be changed, added, or lost, causing mutation. Mutations can possibly render the protein useless.